Okay, let's start. Number 10, hemlock, the poison that killed a philosopher while he kept talking. A man drank a cup of liquid and stayed fully conscious while his legs went numb, then his arms, then his chest, right up to the moment his heart gave out. He never lost the ability to speak. In 399 BCE, the Athenian court sentenced Socrates to death by hemlock, and Plato's account in the Phaedo describes a death that took roughly 30 minutes, the paralysis climbing the body from the feet up while the mind stayed intact almost to the last breath. Athens used hemlock as its standard method of executing citizens. It came from Conium maculatum, a member of the carrot family that grows wild across the Mediterranean and looks, to anyone who hasn't studied it, almost identical to parsley or wild fennel. Prison officials mix crushed seeds and leaves into a drink made fresh for each condemned prisoner. Athenian jailers reportedly kept the recipe standardized, grim as that sounds, because a death that dragged on too long or ended too fast for witnesses to register defeated the point. The active compound is conaine, an alkaloid that blocks nicotinic acetylcholine receptors at the neuromuscular junction, the same receptors a nerve uses to tell a muscle to contract. Toxicologist John Mann, in his 1992 book Murder, Magic, and Medicine, traces how the compound works and notes that roughly zero point 0.5 to 1 gram of conaine is enough to kill an adult, a quantity that would fit inside a sugar packet. Conaine never touches the brain directly. It shuts the body down from the extremities inward, while consciousness stays largely untouched, which is exactly why Plato's account reads the way it does. A man narrating his own paralysis as it happens. The lethal threshold sits below the weight of a children's aspirin tablet, and death usually comes from respiratory paralysis rather than any direct effect on the heart. The condemned suffocates slowly while every muscle above the waist has already stopped answering. Before it reached the Athenian court, hemlock was just a roadside weed, common enough that livestock sometimes died from grazing near it by accident. Farmers across Greece and Rome learned to spot it by smell. Crushed hemlock leaves give off a sharp, mousy odor that sets them apart from their edible lookalikes. The plant carried no ceremonial weight before the 5th century BCE. It was a hazard, not a ritual. For the condemned, hemlock meant a death without violence, without blood, and without a mark left on the body, and that was the whole point of choosing it. The Roman writer Pliny the Elder, in his Natural History from around 77 CE, describes hemlock as a favored method among elderly citizens who chose to end their own lives rather than face a slow decline, a use separate from its role as a tool of the state. The plant offered something rare among poisons, a death that looked to onlookers less like murder and more like sleep arriving early. Athenian law required the condemned to drink the cup himself rather than have it forced on him, and that detail turned the execution into something closer to a ritual of compliance than an act carried out by someone else's hand. Hemlock still grows along roadsides and riverbanks across Europe, North America, and parts of Asia, and it still looks like a dozen harmless relatives to anyone who hasn't been taught the difference. Poison control centers across Europe and North America record fatal misidentifications every year, foragers mistaking it for wild carrots or parsley, most of them with no idea they've picked up a 2,500-year-old execution method. The plant that killed Socrates never stopped growing. It just stopped being feared. The court that killed Socrates didn't have to manufacture anything. The weed was already growing outside its own walls. 6,000 kilometers away, in the forests of the Amazon basin, killing took a different kind of skill altogether, one built not on identification but on preparation. Number 9. Batrachotoxin, the frog poison so potent a single animal could arm an entire hunting party. 2 micrograms is enough to stop a human heart. That's a speck of powder smaller than a grain of table salt, and it comes off the skin of a frog roughly the size of a thumbnail. The golden poison frog, Phylobate terribilis, native to the rainforests of Colombia's Pacific coast, carries enough batrachotoxin in its skin secretions to kill around 20 adult humans, or 2 African bull elephants from a single frog. The Imbera and Noanama peoples of western Colombia built a hunting technology entirely around this animal, using it for generations before any outside chemists figured out what the poison even was. The frog lives right out in the open on the forest floor in broad daylight, unlike most toxic or venomous animals, which lean on camouflage or nocturnal habits instead. Its bright coloring already does the job of a warning sign, one that predators learn to respect after a single encounter. Hunters would press a dart tip against the frog's back or, according to some documented accounts, skewer it on a stick and roast it briefly over a fire to draw the toxin out through the skin, coating dozens of blow dart tips from one animal in the process. The prepared darts stayed lethal for up to 2 years. A hunter could bring down a monkey or a bird high in the canopy with a dose measured in micrograms. Because batrachotoxin doesn't need mass to work, it needs precision. Batrachotoxin locks open the sodium ion channels in nerve and muscle cells, the microscopic gates that control the electrical signal behind every heartbeat and muscle contraction. Once those channels are forced open and held there, the cell can't reset itself. Pharmacologist John Daly at the National Institutes of Health spent decades studying the compound after he began collecting frog specimens in Colombia in the 1960s, and his research, published across several papers through the 1970s and 1980s, established that batrachotoxin ranks among the most potent naturally occurring toxins ever isolated, roughly 10,000 times more potent by weight than cyanide. A dose that size, absorbed through a cut or puncture wound, reaches the bloodstream fast enough that a struck monkey can drop from a branch within minutes, instead of escaping wounded into the canopy. The frog doesn't make the toxin itself. Daly's later fieldwork showed that captive-bred golden poison frogs raised without access to their native diet of toxic beetles and mites lose their toxicity entirely within a few years. The frog is really just a vessel, one that concentrates a chemical weapon first assembled by the insects it eats. To the Imbera, the frog worked something like a natural armory rather than a curiosity, a renewable resource that turned foraged insects, filtered through an amphibian's metabolism, into weapons capable of dropping prey many times the hunter's own size. The forest supplied the poison. The hunter just had to know where to find the right frog. A single golden poison frog, about the size of a large coin, could arm enough darts for several hunting trips before its skin secretions needed to build back up. The rainforest handed the Imbera a weapon they never had to build themselves. Across the Mediterranean, roughly 2,000 years later, an empire built its poisons in kitchens instead, using nothing more exotic than rock and metal pulled straight out of the ground. Number 8. Arsenic, the element that made poisoning an imperial profession. A Roman emperor's food taster died first, then the emperor himself, and the physician who prepared the meal walked away with a fortune paid by the emperor's own wife. In 540 CE, the Roman historian Tacitus records that Agrippina the Younger arranged the death of her husband, Emperor Claudius, with a poison supplied by a woman named Locusta, a convicted poisoner Agrippina had freed from prison specifically for the job. Locusta went on to work as a paid poison consultant to the imperial court for years afterward, her services treated openly enough to count as a professional trade rather than a crime she had to hide. According to Suetonius, Emperor Nero later granted her an estate and gave her students to train, turning what had started as one contracted killing into something close to a state-sponsored profession. Arsenic trioxide, refined from arsenic-bearing minerals like arsenopyrites and orpiment, was cheap, colorless, and nearly tasteless mixed into food or wine, which is why it dominated poisoning in the ancient and medieval world for well over 1,000 years. Miners across the ancient Mediterranean and later medieval Europe pulled it out as a byproduct of smelting other metals. So the raw material behind the era's most infamous poison was for centuries just industrial waste sitting next to any working copper or silver mine. The compound blocks pyruvate dehydrogenase, an enzyme cells need to convert food into usable energy, and death usually follows within hours to days depending on the dose, preceded by vomiting, severe abdominal pain, and organ failure that looked a lot like dysentery or cholera, both common enough in the ancient world that a poisoning could pass without anyone noticing. Toxicologist John Trestrail, in his 2007 book Criminal Poisoning, documents just how much arsenic dominates the historical poisoning record, precisely because its symptoms were so easy to mistake for natural illness, a confusion that lasted until reliable detection methods showed up in the 19th century. Chemist James Marsh developed a dependable arsenic test in 1836 that finally let courts tell a poisoning apart from a natural death with any real confidence. Locusta wasn't some strange exception. She was the product of demand that Roman elite society had already normalized. Political assassination by poison happened often enough within the imperial court that historians Suetonius and Tacitus both write about it as a routine hazard of being close to power, right alongside more direct forms of violence. The poison left no wound, needed no weapon, and could be handed over by anyone with access to a kitchen, a slave, a servant, a spouse. What made arsenic dangerous wasn't the chemistry alone. It was a century of medical ignorance surrounding it, a gap that let killers hide inside the ordinary background noise of disease and death in a world without autopsies. A body buried after an arsenic poisoning could sit in the ground for decades and still turn up a detectable trace, because the element binds permanently to hair and nail keratin, a fact that eventually caught up with poisoners whose victims got exhumed generations later. Locusta worked with whatever a mine and a mortar could provide. In those same centuries, on the other side of the world, hunters in the Amazon basin were working with something no mine could ever produce, a plant that could stop a human heart within minutes, while leaving the meat of the animal it killed perfectly safe to eat. Number 7. Curare, the poison that paralyzes everything except the mind, and is harmless if you swallow it. An animal shot with this poison stays fully aware while its muscles shut down one by one, unable to move, unable to breathe, its heart still beating right up until the moment it can't. And the hunter who killed it could eat the meat afterward without any risk at all. Curare covers several related plant-based preparations that dozens of indigenous groups across the Amazon and Orinoco basins developed independently, and it only works once it's in the bloodstream, through a wound, a dart, or an arrow tip. Preparation methods varied enough between groups that some historical accounts describe 3 distinct categories based on what container held the finished paste: pot curare, calabash curare, and tube curare, each tied to a different regional tradition and, according to some sources, a slightly different plant recipe. Swallow it though, and it breaks down harmlessly in the digestive tract before it ever reaches the muscles it's meant to paralyze. The German explorer and naturalist Alexander von Humboldt documented curare preparation among the Piaroa and other groups along the Orinoco River during his 1799 to 1804 expedition, describing a process that could stretch across several days and involved boiling down the bark and stems of vines from the genus Chondrodendron and Strychnos into a thick, dark paste. The active compound, later isolated and named D-tubocuranin by 20th century chemists, blocks the same nicotinic receptors that hemlock's conine targets, but through a different mechanism. Instead of activating the receptor, it physically sits on the site and blocks it, so the nerve signal never reaches the muscle at all. The prey animal doesn't lose consciousness. It simply loses the ability to respond to its own brain's commands, muscle group by muscle group, until the diaphragm stops moving and breathing ends. For hunters across the Amazon basin, Curare solved a problem that arrow force alone couldn't fix. A blow dart or light arrow rarely carries enough kinetic energy to kill a monkey or bird outright through blood loss or trauma. Curare made the weapon's own force beside the point. A scratch was enough, and the poison finished the job within minutes, letting hunters take down prey from high in the canopy with weapons far too light to do it otherwise. A blow dart weighing only a few grams, tipped with curare paste as thick as tar, could paralyze a howler monkey weighing several kilograms before it ever hit the ground, dropping cleanly instead of scattering wounded through the branches. The same compound that let a hunter fell a bird with a splinter-sized dart would later let a surgeon operate on a living, breathing patient. Anesthesiologist Harold Griffith used purified curare during a 1942 surgery in Montreal to bring on controlled muscle paralysis, a technique that became standard in operating rooms worldwide within a decade. A poison perfected in the rainforest canopy for killing prey without wasting the hunter's arrows, 4 centuries later became one of medicine's essential tools for keeping a patient's body still enough to save their life. The difference between the two uses came down almost entirely to dose and support. A surgical team keeping a patient breathing on a ventilator turned the very same paralysis that once dropped a monkey out of the canopy into a controlled, survivable state. The Amazon's answer to a weak arrow was a poison invisible in the meat it killed. Across the Mediterranean, an empire's answer to political rivalry was a poison just as invisible in a cup of wine. One brewed not from a vine, but from a plant so ordinary it grew in gardens meant for something else entirely. Number 6. Aconite, the queen of poisons that armed warriors long before it armed assassins. A single leaf, absorbed through a small cut in the skin, could kill a grown man before symptoms even had time to fully develop. Aconite comes from the plant genus Aconitum, known variously as wolfsbane, monkshood, or the queen of poisons, and it carries aconitin, an alkaloid strong enough that ancient hunters across Asia and Europe were coating arrowheads and spear tips with it centuries before it became a favorite tool of court intrigue. The plant's hood-shaped purple flowers, which give monkshood its name, grow across temperate mountain meadows in both Europe and Asia, so two cultures on opposite ends of the continent stumbled onto the same weapon inside the same genus of flower, independently of each other. In ancient China, aconite root, called fuzi or wuto depending on how it was prepared, shows up in hunting practices recorded as early as the Han Dynasty, used to tip arrows for taking down large game when a clean kill through blood loss alone couldn't be counted on. In ancient Greece, historical accounts gathered by pharmacologist and toxicologist John Mann note that Greek hunters used aconite-tipped weapons specifically against wolves, which is probably where the plant's common name, wolfsbane, comes from. Aconitin binds to voltage-gated sodium channels in nerve and heart tissue, similar in principle to how batrachotoxin works, but structurally different, and it locks those channels into a state of constant activation. The result is a heart that beats erratically and then stops, along with numbness that usually starts at the point of contact and spreads outward, often within 20 to 40 minutes of a lethal dose. As little as 2 milligrams of pure aconitin, less than the weight of a grain of rice, is enough to kill an adult, which is why ancient hunters coating arrowheads worked with the plant's raw sap rather than any purified form. They trusted quantity over precision. Aconite's danger wasn't limited to arrowheads. The plant grows as an attractive purple-flowered garden ornamental across temperate parts of Europe and Asia, and its root has been mistaken for edible tubers like horseradish or Jerusalem artichoke, often enough that toxicologists still log fatal misidentification cases today. The Roman naturalist Pliny the Elder specifically warned readers not to plant aconite near vegetable gardens, treating its toxicity as a household hazard rather than a battlefield one, which sets it apart from most of the other entries here. It killed by accident just as often as by design. What aconite gave ancient hunters was reach without exposure. A warrior or hunter didn't need to overpower an animal or an enemy up close. A shallow wound from a treated blade did the work that raw strength alone couldn't guarantee, and the poison acted fast enough that a wounded target rarely made it far before collapsing. Physicians serving both the Roman and later Chinese courts are recorded testing food for a bitter metallic aftertaste tied specifically to aconite poisoning, a precaution routine enough to shape court dining customs for centuries. A weapon built for wolves and enemy soldiers eventually found its way into royal courts as a tool for quietly killing rivals. But not every ancient poison needed a blade. Some killed through nothing more than a bite of an insect small enough to sit on a fingertip. Number 5. Cantharidin, the beetle extract that killed as often as it seduced. A powder made from crushed beetles could kill a man outright, or in a dose small enough to miscalculate fatally, get sold to him as a way to seduce someone else. Cantharidin, extracted from the blister beetle, most notably the species Lita vesicatoria, commonly called Spanish fly despite not being a fly at all, was one of the ancient Mediterranean world's most dangerously two-faced compounds, marketed as an aphrodisiac while quietly feared as a poison, often by the very people selling it. The beetles were harvested by hand across olive and ash groves in southern Europe, dried and ground into a fine powder, a process recorded in ancient Mediterranean medicine long before any chemist pinned down cantharidin as the exact compound behind both effects. Roman physician and encyclopedist accounts collected in Pliny the Elder's Natural History describe cantharidin's blistering effect on skin and its use, in controlled doses, as a treatment for warts and other skin lesions. The compound irritates and blisters any tissue it touches, and swallowed in a large enough quantity, it causes severe internal blistering of the gastrointestinal and urinary tract, followed by kidney failure and, often, death. The gap between a therapeutic dose and a lethal one was narrow enough that ancient physicians prescribing it, whether or not they fully grasped the risk, were operating close to poisoning territory with every dose they measured out. A dried, crushed dose lighter than a modern paperclip could take a patient from mild irritation to fatal kidney damage, with almost nothing about the powder's color or texture to warn whoever was measuring it. One case in the historical records cemented the compound's split reputation for good. The first-century Roman writer Suetonius records that Livia, wife of Emperor Augustus, allegedly used cantharidin-laced food at social gatherings to provoke embarrassing behavior from guests she wanted to discredit, using the compound's genital irritation as a form of political humiliation rather than an outright killing. Whether or not that particular story holds up, it reflects a wider understanding at the time. Cantharidin sat somewhere between weapon and social tool, useful for control in ways that didn't always require a body. Modern toxicology puts cantharidin's lethal dose in humans at roughly 10 milligrams, an amount smaller than a single grain of rice, based on case studies forensic toxicologists have collected throughout the 20th century, documenting accidental death from folk aphrodisiac preparations. The compound doesn't tell the difference between its advertised use and its lethal one. The dose that made someone uncomfortable and the dose that killed them sat close enough that the ancient world's aphrodisiac trade functioned in practice as an unregulated poison market operating in plain sight. Cantharidin blurred the line between seduction and assassination at crowded Roman dinner parties. On the other side of the ancient world, a different poison blurred an even stranger line, the one separating medicine cabinet from murder weapon. Number 4. Tetrodotoxin, the poison that could make a living man look dead. Doctors in 18th century Japan documented cases of men who ate the wrong part of a fish, collapsed within 30 minutes, and appeared completely dead, no pulse a hand could find, no visible breathing, while staying fully conscious inside a body that could no longer move or signal that anything was wrong. Tetrodotoxin, concentrated in the liver, ovaries, and skin of pufferfish, known in Japan as fugu, ranks among the most potent naturally occurring nerve toxins modern toxicology has ever studied, roughly 1,200 times more toxic than cyanide by weight. Shell middens along the Japanese coast show that communities were eating pufferfish and presumably dying from it now and then as far back as the Jomon period, thousands of years before any formal licensing system existed to catch a chef's mistake. The compound blocks voltage-gated sodium channels in nerve cells, the opposite of what aconitine does, since aconitine locks those same channels open. Tetrodotoxin seals them shut instead, so no nerve signal can fire at all. The result is a paralysis that spreads from the extremities inward, eventually reaching the diaphragm, while consciousness and awareness usually stay completely intact until the very final stage. There's no antidote. Survival depends entirely on artificial respiration, keeping the body oxygenated long enough for the toxin to clear the system on its own, a process that can drag on for several days. A single pufferfish liver, smaller than a fist, can hold enough tetrodotoxin to kill upward of 30 adults. Japanese chefs who train specifically to prepare fugu have worked under formal licensing requirements since the Japanese government regulated the practice in the 20th century, precisely because the margin between a delicacy and a fatal dose sits inside a single organ that has to be removed with total precision. But tetrodotoxin's most documented use as a deliberate weapon comes from an entirely different tradition. Anthropologist Wade Davis, in fieldwork he carried out in Haiti in the early 1980s and later published in his book, The Serpent and the Rainbow, investigated preparations used in certain Haitian voodoo practices that incorporated pufferfish-derived tetrodotoxin, documenting how a survivable but severe dose could bring on a state of paralysis convincing enough to pass for death to observers who didn't know the toxin's particular effects. What set tetrodotoxin apart from almost everything else on this list wasn't lethality by itself, it was the toxin's ability to counterfeit death with enough precision to fool people standing right over the body. No detectable pulse, breathing shallow enough to go unnoticed, skin cool to the touch. A poison that killed through paralysis was dangerous enough on its own. A poison that could fake a corpse changed what death itself could be used to accomplish. A fish that could counterfeit a corpse belonged to the sea. The next poison belonged to the garden. A plant so ordinary that Roman women reportedly used it as cosmetics, dabbing onto their own eyes the same compound that in a slightly larger dose could stop a heart.
Number 3. Belladonna, the beautiful lady poison that Romans wore on their faces. Roman and Renaissance women placed drops of a plant extract directly into their own eyes to dilate the pupils, chasing a fashionable doe-eyed look, using the same compound that, in a slightly larger dose, ranked among antiquity's most reliable ways to kill someone. Atropa belladonna, deadly nightshade, takes its species name straight from this cosmetic use. Belladonna means beautiful woman in Italian, a label applied centuries before toxicologists understood what the plant's active compound actually did to the human nervous system. The plant produces atropine and related tropane alkaloids that block acetylcholine receptors throughout the body, a different target than the ones hemlock and curare interfere with, but with effects that ripple across nearly every organ system. It grows along woodland edges and disturbed ground throughout Central and Southern Europe, favoring the same shaded, nutrient-rich soil that made it easy to grow deliberately in a household garden without raising suspicion. In small doses, atropine dilates the pupils, dries the mouth, and speeds up the heart rate, which was exactly the cosmetic effect Roman and Renaissance women were chasing. In larger doses, it causes delirium, hallucinations, dangerously fast heartbeat, and eventually respiratory failure. Every part of the plant is toxic, but the berries, dark, glossy, and reportedly sweet-tasting according to historical poisoning accounts, pose the greatest risk to accidental victims, children especially, since nothing about how they look signals any danger. As few as 2 to 5 berries, an amount a curious child could gather in one closed fist, can deliver a fatal dose. The Roman historian Tacitus, in his Annals, recounts the poisoning of the young Roman general Germanicus in 19 CE.
an incident where his contemporaries suspected deadly nightshade among the possible agents, part of a wider pattern of suspected political poisonings within the early imperial court. Alongside the arsenic-based methods, figures like Locusta practiced a generation later. Ancient physicians, including the Greek physician Dioscorides, writing in the 1st century CE, documented belladonna's toxic effects in his pharmacological text De Materia Medica, cataloging symptoms of poisoning that modern toxicology confirms right down to the detail. The same delirium, the same fixed dilated pupils, the same racing pulse he wrote about nearly 2,000 years ago. Physicians treating belladonna poisoning today still lean on a mnemonic that dates back generations, describing the symptoms as blind as a bat, mad as a hatter, red as a beet, hot as a hare, a description of dilated pupils, delirium, flushed skin, and fever that matches Dioscorides' ancient account almost word for word. Belladonna's danger lived inside its double identity. It was cosmetic and lethal, medicinal and murderous, all depending on a margin of dose that ancient users had no reliable way to measure precisely. A woman applying drops to brighten her eyes and a poisoner lacing a rival's wine were drawing from the exact same plant, separated only by concentration and intent. A plant that beautified and killed depending on dose sat quietly in Roman gardens for centuries. The next poison needed no garden and no dose calculation at all, because a single drop of it, applied anywhere on unbroken skin, was already more than enough. Number 2. Ricin, the poison hidden inside an ordinary bean, potent enough to kill with a speck. A grain of purified powder smaller than the head of a pin can kill an adult human. Extracted from a bean so common it was grown across Egypt, the Mediterranean, and the ancient Near East for reasons that had nothing to do with poison, its oil. Ricin, drawn from the seeds of the castor plant, Ricinus communis, ranks among the most toxic naturally occurring substances by weight ever identified, with a lethal dose for an adult measured in micrograms per kilogram of body weight, small enough that just a few purified milligrams counts as a fatal amount. The castor plant grows readily across warm, dry climates from North Africa through the Middle East and ancient South Asia, producing seed pods and clusters a farmer could harvest by the basketful, with no specialized equipment at all. The castor plant grew widely across ancient Egypt, where archaeological evidence and papyrus records confirm castor oil was pressed from the seeds for lamps and for early medicinal and cosmetic preparations, a use that goes back as far as pre-dynastic Egyptian sites and shows up in the Ebers Papyrus, one of the oldest surviving medical texts, compiled around 1550 BCE. The oil itself, once pressed and refined, isn't toxic. Ricin is a protein that stays bound to the leftover seed mash after pressing, so the plant's two uses, one harmless and widespread, one lethal and concentrated, come from the exact same harvest, split apart only by a processing step ancient Egyptians had no particular reason to bother with on purpose. Ricin disables ribosomes, the cellular machinery every living cell relies on to build proteins, which effectively shuts down a cell's ability to sustain itself from the inside out. Once enough cells in a vital organ lose that function, organ failure follows within days. A single ricin molecule, once inside a cell, can disable roughly 1,500 ribosomes before the cell finally dies, part of why the compound stays lethal at doses too small to see without magnification. Unlike arsenic or aconite, ricin's action is slow and cellular rather than immediate and neurological, so a victim can survive 1 to several days after exposure while symptoms steadily worsen before death, a timeline that made ricin poisoning hard for ancient observers to trace back to any specific meal or moment of exposure. What made ricin dangerous wasn't sophistication. The compound sat, essentially invisible, inside the waste product of one of the ancient world's most common agricultural crops, needing no specialized cultivation, no rare import, no exotic hunting trip into unfamiliar territory. A field of castor plants large enough to keep a village stocked in lamp oil for a season also held in its discarded seed mash enough raw material to poison every person who ever walked past it. Anyone with access to castor seeds and the patience to process them had access to one of the deadliest substances the ancient world would ever come across, whether or not the people living around that plant had any idea what they were standing next to. You've probably used a cosmetic or a laxative containing castor oil without a second thought, because the toxic protein and the useful oil separate cleanly enough that one has stayed a shelf staple for 4,000 years, while the other became shorthand for undetectable murder. 9 entries in, and the poisons kept building on each other. One plant, one beetle, one fish. The final entry didn't lean on a single organism at all. It combined several, on purpose, into something no accident of nature would ever have produced by itself. Number 1. The Scythian arrow poison, a recipe so lethal that a scratch was a death sentence. An arrow wound that drew barely a teaspoon of blood could still kill a fully grown, healthy warrior within days through infection so severe and so specific that ancient physicians describe symptoms unlike anything the wound itself should have caused. Not the arrowhead, not blood loss. The Scythians, a nomadic warrior culture that dominated the Eurasian steppe from roughly the 7th century BCE through the 4th century BCE, built one of the ancient world's most sophisticated biological weapons out of ingredients no army before them had thought to combine. Their whole society trained in mounted archery from early childhood, and Greek accounts describe entire war bands carrying quivers stocked with treated arrowheads as standard equipment, not some specialized tool kept aside for assassination. Ancient accounts collected by the Greek geographer Strabo, along with the toxicological writings of the Roman-era Greek naturalist Aelian, describe a Scythian poison preparation later scholars sometimes called Scythicon, made from venomous snake matter, most likely the steppe's local vipers, mixed with decomposing human or animal blood, and according to some ancient accounts, feces, left to rot together before being applied to arrowheads. Historian Adrienne Mayer, in her 2003 book, Greek Fire, Poison Arrows, and Scorpion Bombs, reconstructs the chemistry behind this recipe and argues the mixture worked through 2 mechanisms stacked on top of each other rather than just one. Snake venom's tissue-destroying enzymes delivered straight into the bloodstream, combined with a heavy bacterial payload from the putrefied matter that seeded the wound with pathogens capable of causing tetanus and severe gangrenous infection, even in cases where the venom dose alone wouldn't have killed outright. That's what set the Scythian weapon apart from a simple venomous dart. Snake venom on its own breaks down fairly quickly and loses potency once it's out of a living animal, which is why most venom-based weapons had to be prepared fresh and often. The Scythians got around that limit by essentially engineering a compound weapon. Venom for the immediate toxic hit, bacterial contamination for a secondary infection that could kill days or weeks later, even if the first dose failed, and putrefied tissue that, according to Mayer's analysis, likely also seeded the wound with the Clostridial bacteria behind tetanus, a disease ancient physicians recognized by its telltale muscle spasms and lockjaw, but had no way to treat. A warrior who made it through the initial wound could still die from infection a week later, far from the battlefield, in a way no ancient physician could ever trace back to the original scratch. Herodotus, writing in the 5th century BCE, describes Scythian warriors as expert horse archers, whose reputation for lethal, near-mythical accuracy across the steppe made them one of the most feared military forces the Persian and Greek worlds ever ran into. Even a wound that missed every major organ still came with the same eventual sentence, and the poison arrow tradition Mayer's research reconstructs offers a solid chemical explanation for how a culture without metallurgy or fortifications on the scale of its rivals still managed to hold territory against far larger empires for centuries. The weapon didn't need overwhelming force behind it. It just needed a wound that would kill no matter how the battle itself turned out. A single treated arrowhead carried in a quiver alongside dozens of untreated shafts turned an otherwise survivable graze into a mortal injury days before anyone would normally think to treat the wound as serious. The Scythians never had to beat an enemy outright in a single engagement. They just needed to touch him. Every other poison on this list worked through a single mechanism, refined and aimed at one kind of target: a heart, a nervous system, a set of receptors. The Scythians built theirs to fail the body in multiple ways at once, so that even a warrior who walked off the battlefield carried the battlefield home with him in his blood. 10 poisons, 10 solutions to the same ancient problem: how to kill without ever needing to win a fair fight. A garden, a beetle, a fish, a corpse left to rot on purpose. None of the ingredients were exotic. What set the people on this list apart from everyone else standing in the same forest or the same market was a willingness to look at something ordinary and ask what it could do to a body. Each one worked by exploiting something the human body had no defense against: a receptor it couldn't shut off, a wound it couldn't sterilize, a dose too small to taste. None of these compounds needed an army, a fortress, or even physical strength behind them. They just needed someone patient enough to figure out what a plant, an animal, or a corpse could already do, and willing to aim it at another person. The chemistry hasn't changed since. Every mechanism described here, blocked receptors, seized ion channels, disabled ribosomes, still run in a hospital pharmacy today, just refined into medicine instead of murder. If you want to see what else the ancient world weaponized long before it understood why any of it worked, click the video on screen.